Hi guys, welcome to week 14 of BIO113. This is our last lab of the year before the final exam. So uh, what we're going to talk about this week is water quality. And there's a nice, beautiful picture of some water. It looks really nice and pristine. But uh, you shouldn't just go drinking any of this water in nature. It can contain any kind of organism that might make you sick. And so we're going to talk a little bit about water quality in general here. So water. Water covers approximately 70% of the Earth's surface, as you may have heard. But of that 70%, 97% of that's down in the oceans. And that's basically salt water, undrinkable. About 2.4% of that is in the polar ice caps. That number may have shrunk since I first made this slide because of global warming or global climate change. But again, undrinkable because it's frozen water. So the only sources we have of water are 0.6% of all the world's water supply is in rivers, lakes, and other surface waters. And that's not a large amount, so you can imagine water pollution is a big problem when it comes to having water, which we need for life to exist. All life requires water. So we need to protect our water supply. So when we talk about water quality, we're going to talk about each one of these bullet points in looking at and determining the quality of the water. And not just site description, but all of these things are important in determining how good the water is or how what level of quality the water is. All right, so site description in general, you can just look at a site and tell if it's healthy or not most of the time, at least based on the way it looks. Is there, is how deep is the water? If it's a stream, how fast is the flow rate? Obviously, a slow flow rate is going to allow for more things to grow in the water. Maybe like what's the substrate? So what is the bottom of the stream like? What's the shoreline vegetation look like? Does it look healthy? Does it look like it's growing well? And pollution, is there a lot of trash around? So all these things can affect your water quality and not only the water quality, but the things that grow in the water. So as I said, substrate is the sediments and rocks that make up the bottom of the water, where the water is. So you have boulders, cobbles, gravel, and sand. And even like around here, a lot of the times you'll see limestone. If you look at some of the streams around here, and a stream with limestone substrates would have a higher or lower pH. Well, let's think about this. So limestone is calcium carbonate, and when limestone gets broken down, it's more, you get it broken down into calcium and carbonic acid or something like that. So the pH would go lower because acids are lower. So uh, that's just a side note. This won't be on the exam. Just to give you an idea, now it's not a huge difference, but the pH here of water may be lower than it is in other places. All right, so site description. It's pretty obvious here which one of these has more oxygen in the water. You can see on the right here, we've got a nice flowing stream. It's got large boulders, and you can see these boulders are help cause rifts of roils in the water, and basically adding oxygen to the water. Whereas on the left is the very stagnant looking pond filled with garbage. It does not look healthy at all. I think I would not swim in that. I would probably step into this stream over here, but definitely not this water on the left. So site description these are two extremes, but it can help you determine water quality from the just first glance. Temperature. So temperature has a big effect on water quality as well. Temperature can affect the things that are living in the water. So organisms that live in water usually grow at a specific temperature that they're used to. So streams are usually cooler than the atmosphere, and fish and other organisms rely on this temperature to be stable. Some organisms actually rely on higher temperatures to grow. So you'll see things like bacteria can grow when the water warms up to a certain temperature. And you can see here is an example of, you've got a power plant that uses water from the river here to cool off its whatever they do in power plants to cool it off. Uh, and then it just spits that water back out in the river, which you think is no big deal. But if you look at the thermal image here on the right, you can see where the water goes back into the river. You can see the red indicates hotter water, and it slowly dissipates, but it creates a broad area of warmer water here in the stream. And this can really affect how, fish can't grow in this area because it might be too hot. Maybe E. coli or bacteria can grow in here because it's warmer, it can grow better. So you can see thermal runoff is what we call that, how that can affect an ecosystem quite a bit. And pH, we talked about pH just briefly, but pH is important for organisms or plants or animals to live. Most things like a neutral pH to survive, or around neutral, usually 6 to 8 pH range. So pH, of course, the more acidic it is, anything 1 to 6.9 is considered acidic. 7 is considered neutral, and basic is 7.1 to 14. So pH can have a great effect on enzymes, as we've studied previously. In this semester, we looked at enzymes and how they can be affected by different things. And one of the things we looked at was pH. And having an enzyme in the pH that's not its normal pH can really affect whether or not it can function. And increased or decreased pH compared to what's normal can make toxins or nutrients more or less available depending on the organism that's in the environment. So pH can have a great effect on what can survive in certain environments. All right, conductivity. So how can conductivity affect water quality? So what conductivity is basically water's ability to conduct electricity. And it's not really necessary for water to conduct electricity in the natural setting, but what you can tell by its ability to conduct this electricity is basically the concentration of ions in the water. So salts or limestone or things like that. And this is measured in what's called MHO per cm per centimeter, or Siemens. This German guy named Siemens. But just on a side note, MHO is ohm backwards, since ohms is what you measure resistance in. MHOs is what you measure conductivity in. So it's a reciprocal. It's the inverse of resistance. So distilled water is water that you have basically taken almost all of the ions out of. Its conductivity levels range from 0.5 to 2 Siemens. Mineral or hard water ranges from 100 to 1,000 Siemens. And industrial waste can be up to 10,000 Siemens. Now, I used to have a note on here. Well, we'll talk about hardness later on, I think. Okay, so back conductivity just measures the amount of dissolved ions in the solution. And ions can be important for the things that live in the water. Not much can live in distilled water because, as we remember from our, we had a lab on plants, and in that lab we had an experiment where we looked at plants in salt water versus fresh water, and also talked about it being in distilled water. So if a plant grows in a solution that's normally 1% salt or ions, and you put it in distilled water, it's going to gain water across the membrane because it's trying to balance it out with the outside, and the outside has very low ions. So that cell is going to swell up and become turgid, right? If it's got too much ions on the outside, then the water is going to leave that cell and it's going to shrink and become plasmalized. Okay, so conductivity can affect plant and animal growth in that manner. All right, dissolved oxygen. So oxygen is very important for organisms that live in the water, much like a lot, you think of fish, they don't breathe like you or I. What they do is they take water in and run it over their gills and pull the oxygen out of the water that way. And so the more soluble water in the water, the easier it is for them to pull oxygen out of that soluble water, or out of that water. So oxygen, of course, is soluble in water. And as you increase the temperature, you have less oxygen in the water. And then as you decrease the temperature, so colder water temperatures, you get more oxygen in the water. Now, obviously, if you freeze the water, there's not going to be much oxygen. Well, it'll still be there, but it's not accessible. So the recommended dissolved oxygen concentration for fish, and fish is usually a good measure of water quality, how well the fish are doing. For warm water fish, you want something that's a little less than 4 milligrams per liter, not much, but around 4 milligrams per liter. But for cold water fish populations, it should be more than 5 milligrams per liter of dissolved oxygen. Now, you can get large groups of fish die. Fish kills occur when this dissolved oxygen drops below this 4 milligrams per liter. And you can see this quite a bit. Obviously, if you had that stagnant water we looked at earlier, you probably would not have any fish living in there. They would all die because the oxygen levels were just too low. Okay, so now hardness, and this kind of relates to that conductivity a little bit. Hardness, though, is a measure of calcium and magnesium ions that come from calcium carbonate. So hardness can range from 0 to hundreds of parts per million, and usually you'll hear things measured in parts per million, and that's you think, oh, parts per million, that's one part in a million parts. That can't be that big of a number, but a slight difference here can have an effect on what can grow in the water or survive in the water. So soft waters contain small concentrations, and I don't know, you probably don't experience that around here, because what we have is hard water, because we have high concentrations of calcium and magnesium in our water, mainly because most of this area is covered in limestone, and limestone dissolves to get into the water, and that's where you get our calcium carbonate, which is what limestone is in the water, makes it harder. So drinking water standards are 0 to 60 parts per million is soft water, 60 to 120 is medium, 120 to 180 is hard, and over that is very hard. And if you look down here, just I got this number from 2016, the average parts per million in Lexington is 155 parts per million, and it ranged from 58 to 400 parts per million. So you can see we have hard to very hard water here in Lexington, meaning you have a lot of dissolved calcium carbonate ions in the water. All right, so nitrates. So nitrogen is a key element for plant growth, obviously, but too much nitrogen, too much of anything is bad. And too much nitrogen gets in your water supply, it's a big problem. So you can have sewage or too much fertilizer or waste water can cause nitrates to go up. You like to see about this time of year, people will fertilize their lawns with lawn fertilizer or things like that, but people tend to put more than they really need on their lawns. And what happens with that excess is when it rains, it just washes right down to the sewer drain and goes into the water supply. And so plants that are, that grow in that water get a large amount of nitrogen coming in. And we'll talk about how this can affect the water quality in a second. But nitrates and phosphorus can stimulate unwanted growth of algae. Okay, you have algae growing in most waters anyway, but too much algae can be bad. And too much nitrates in drinking water can be harmful to young children and unborn children. The maximum contaminant level permitted in drinking water is 10 parts per million. And as according to, I get most of these stats from the Kentucky American Water Company's website, Lexington was 0.12 to 0.34 parts per million, so well under the maximum standard for the United States. And phosphates, we just mentioned those. Phosphorus, again, is important for plant growth, aquatic plant growth especially. You want less than 1.1 part per million unless the water has become polluted from wastewater, then you're going to get higher numbers than that. And excessive phosphorus can lead to what we call eutrophication. So nitrogen and phosphorus can both, too much of that can lead to eutrophication. So here's your normal lake on the left, and you get this algae that grows in there normally, but when you get a lot of phosphorus, excess phosphorus and nitrates, then this algae grows, and basically it looks like it's completely covered the lake. So I want to delve into eutrophication just a little bit more here. So what happened? Here's the process. You've got, normal plants live in the water here. Phosphorus and nitrogen comes from runoff into the lake and raises the levels of nitrates and phosphates in the water. Well, this basically works as fertilizer. As you know, it works as fertilizer on your lawn. Well, it also works as fertilizer on all plants, and algae is no exception. So the more phosphorus and nitrogen that gets in the water, the more these plants can grow, the algae can grow. And of course, sunlight, plants need sunlight. So you get that combination of water, sunlight, and these nutrients. You get algae growing to an extent where it grows so much, like in this picture, it basically covers the entire lake, right? And if you cover this entire lake with a lawn of algae, you actually will reduce the amount of light that gets in to the other parts of the lake. And so what happens here is, because sunlight can't get past this algae that's on the top, these plants that normally grow underwater that normally help add oxygen to the water supply, these guys die off because they can't get sunlight. So it reduces your submerged aquatic vegetation. And when that happens, there's less oxygen in the water. And when there's less oxygen in the water, you can see your fish die off too, or living things that usually require water or oxygen to survive will die off because of the depleted oxygen. So it's a process that basically leads to fish dying because this algae blooms, cuts off sunlight, causes the plants under that to die, which causes oxygen to get depleted in the water, and fish and animals living in the water can die off. All right. Some animals die due to lack of oxygen. And it's terrible. And that's why too much phosphorus or excess phosphorus and nitrates in fertilizers can be a really bad thing. And so just think about that next time if you fertilize your lawn. Now, biological profile. This is easily one of the easiest ways to look at the quality of the water you're looking at. So you always want to see the number of organisms. And you might think that, you're looking at a stream and there's nothing in there, but there's all kinds of organisms growing in that stream and under the rocks, in the water. And the more number of organisms, different kinds of organisms, and the more diverse it is, the better the water quality. Okay. So you got to look at the water, you got to get down and kick up some of the sediments there and look at under that. And then also this riparian vegetation, which is just the plants that grow on the side of the stream, let's say, or the edge of the lake. That's called riparian. And if you have high animal diversity, that's good quality water. Low animal diversity is poor quality. So if you have, you can have a lot of animals, these small animals living there, like crawfish or snails or clams. But if you have just a low variety of these things, but you could have a lot, it doesn't matter how many you have, if you don't have a good diversity, then that indicates that not lots of things can't live there. Only a few things can survive that poor quality. So that's what we're trying to say there with the low animal diversity means poor quality, whereas high animal diversity means many different organisms can survive because it's a good quality environment. All right, bacteria, obviously, uh, some bacteria are really bad for you, and water can be contaminated with bacteria, and more specifically, your fecal coliforms, which are E. coli is one of those. So these are organisms that uh, are found in feces, basically, and feces from animals, humans, and the like can contaminate water supplies. So you want to, when you're looking at water, you can't tell if it's got bacteria in it or not. If bacteria is so small, usually. So you have to take a test sample of the water and look at that. And so what you can do, what we do, test this is we take a sample and put in this broth that has nutrients in there for the bacteria to grow if they're present. And the broth is purple color, and it'll go from a purple to yellow color. It means there is most likely going to be contaminants in there. And of course, once that's seen, you have to do more tests to identify the organisms. So, this would be here we have 3 different samples of water from 3 different sources. And we done all these tests to look at 3 different samples. And, you can look at them and determine, which one of these would be the best quality water, basically. So, if you look at this first one, it's got kind of high levels of phosphates, but not too bad. Hardness is medium, okay, to high, and then no nitrates. It's got good dissolved oxygen, 5 parts per million, and its conductivity is in the mid-range, 173, okay. And pH is perfect, it's 7. Sample 2, it's got very low phosphates, no hardness, no nitrates, very little, high dissolved oxygen, but very little conductivity, and the pH is 5. If I had to guess about sample 2 here, I would probably say that would be distilled water. Because, you can have dissolved oxygen in the water and it's not going to come out with distillation, but you have such low conductivity and low phosphates and no hardness and no nitrates. I would think that would be distilled water there. And, it is not good for you to drink distilled water because it's such low levels of ions. It actually would be much like the plant sample or experiment that we did, where your cells, if you drink deionized water, it would cause your cells to try to take up more water and swell, and basically animal cells actually rupture in that situation. So not good. Now this third sample, it's not a huge difference from the first one, except for you can see dissolved oxygen levels are very low, and that's not good. And your conductivity is very high. So those 2 things would kind of tip you off that it's not a great quality water there. All right, so that's all of the slides that I want to talk about. But now I want to talk a little bit about, so this week we're going to have some homework and get a question set and a quiz that you'll be doing. Let me see, question set here. All right, so this is just going to be your question set. And this question set, I've got also in the lab folder a file that's the water quality lab notes. And you'll see, I've got it way down at the bottom here, but it talks about everything we just talked about with the different aspects of water quality, things that we look at. Basically repeats most of the things I said. There's more information in here than I went over. And it even talks about in detail the experiments you would do to test these things. You don't have to know the experiments for the coming exam, but just be familiar with these different tests and different aspects of water quality, mainly. There is, let's see. Oh, but what I want to get to is in the homework or the question set, obviously it'll talk about, the different water samples and the different levels and how, what can you tell? It's kind of what we talked about in the last slide. And then come down to this one. I talked about this, but you haven't seen this yet. This is the test for coliform bacteria in your water quality. So which of these samples is positive for coliform bacteria. So if you go to the notes section and go near the very end, for some reason it's got stuck at the end. Here we talk about, bacterial profiles, and you go through and it talks in detail about this assay, the purple broth, and how it's this can change color based on what's growing in there. So which of these has positive for coliform bacteria? Well, you can see number 2 is what it looks like originally, so there's no change in number 2. But number 1 and number 3, they're positive. And then the negative is number 2. And then what does the bubble indicate here? That tells you, and you can read it in the note section, but that tells you that there's some anaerobic bacteria there that when they, there's lactose in this for the source of carbon. So when they process this lactose, they give off hydrogen gas, and this is a little upside down, almost like a little tiny test tube in here. And basically, as these bacteria give off this gas, it kind of can get collected in this test tube, and you can see that anaerobic bacteria are present because of that bubble. All right. So if you go on, the next thing you're going to see is there's going to be, I think there's 20 something of these. Let's see how many there are. 20 different invertebrates that live in water. And the idea is that you will identify these. And if you go to the water quality lab notes, let me back up, you'll see, like before, like the last lab in the Animal Kingdom lab, we've got different keys to help identify these things. And so like I said, there's 20 of them. You do your best to identify all 20. At the bottom here is a nice sheet for you to fill in. I only want a common name. I don't need to know if it's sensitive to pollution or tolerant to pollution. If you want, that's fine. I'm not sure if you go to this, it'll talk about, you have to really, this is kind of hard to see, so some of the writing. So just do your best to try to help use these keys to identify the different organisms. All right, and pay attention to what's written out here if you can read it very well. It talks about the different organisms. Okay, and use this to help identify your pictures here. Now, so there's a couple keys, and then there's this third key. Maybe I didn't look hard enough, but I couldn't find anything about tolerance to pollution. So, that's why I'm telling you not to worry about that too much. Okay, just use that to try to identify your invertebrates. Okay, and all these could be found if you were to go out and dig in a lake or a stream. You could find almost all of these guys. Here's a good example. This is a mosquito. I can tell you that from just looking at it. And this is what a mosquito larvae looks like. And this is a lot smaller than it looks in this picture. It's usually about half the size of a regular mosquito there, this larvae. And if you look in some water and you see these, it looks like tiny little things, like trying to squiggle back and forth a little bit. That is the mosquito larvae. So that tells you there's a lot of mosquito getting ready to hatch out of that water. And so that's kind of an indicator of stagnant water, because these larvae can't grow if the water is moving. So you don't get mosquitoes growing from streams. Usually it's from the lakes or ponds and or standing water. So just an idea there. Of course, dragonfly and things like that. So anyway, I found this interesting. You may too. Some of these things you may not have ever seen, and just knowing that they're there, maybe next time you're out looking at the water, you'll identify some of these things. Some are really small. So I know this key over here, one of these keys, it has let me get to it. It's got these lines here. So I think these lines are supposed to be, if you look at the lines, the bar lines indicate relative size. So this bar is supposed to be the same size as this bar, as this bar. You can just imagine they zoomed way out to see that, whereas you zoom way in and it'll be this big. So that is just an indication of like maybe how big certain things are. All right, that's all I have. If you have any questions, feel free to email me, let me know. And like I said, this is the last lab. Next week will be final exam, and it's only going to cover the last half. Everything after the midterm will be on this final exam. Okay. I hope you guys have a good week, and I'll talk to you soon.